Hi, I'm Dom. I'm a PhD candidate at the Burnett Institute and Monash University in Melbourne, Australia. And I'm going to be talking about a modeling analysis that I did with Gavi, the Vaccine Alliance, where we estimated the vaccine, the impact of outbreak response immunization for outbreaks of measles, cholera, yellow fever, meningococcal meningitis, and Ebola. So actually, thanks to Mark, because you've given a great overview of meningitis, which will save me a little bit of time when I get to that part. So as I said, this was an analysis that I did with Gavi. So Gavi funds the creation and the management of vaccine stockpiles, which can be accessed in the event of an outbreak in a country. These stockpiles are managed by groups like the International Coordinating Group for Vaccine Provision or for the Measles and Rubella Partnership. And so what Gavi wanted from us was an estimate of the impact of their historical investment in these stockpiles, because, well, they wanted to understand how much good they had done in the sort of 20 plus years that they've been running, and because they also wanted to be able to motivate future investment. In particular, they are currently developing their Gavi 6.0 strategy, which is going to be their funding strategy from 2025 to 2030. And so hopefully a lot of what we've produced here is going to be useful for informing that funding strategy. So to produce these impact estimates, we had to develop 5 models, those being for cholera, measles, yellow fever, meningococcal meningitis, and Ebola, as I said before. And then we calibrated all of those disease models to a collection of historical outbreak data. Once they'd been calibrated, we produced counterfactual scenarios for each of those outbreaks in which we assumed that no ORI took place. And then by comparing our model simulations in which there was a calibrated baseline with a response against these counterfactuals, we estimated the total number of cases, deaths, disability, adjusted life years, and translated some of those into economic costs to estimate how much had been averted by the presence of the vaccine response across this set of outbreaks. And we also considered what impact has the inclusion of ORI had on the risk of observing large outbreaks. So I'll explain that a little bit more when I get to that slide. But that was just an additional measure that we examined. So in order to develop 5 models in honestly a relatively short amount of time, because Gavi wanted some snappy turnaround on this, we used the StarSim framework, which some of you may know very well. Hopefully some of you will have just heard a bit about from Cliff's talk just before over lunch. But for those of you who don't know, StarSim is an agent-based modeling framework developed by people here at IDM. And its whole thing is that you can flexibly and rapidly develop whatever kind of disease module that you want. And so for our purposes, where we had to build 5 disease models pretty quickly, it was absolutely perfect. And as well, because we wanted to be sort of like aggregating the results and comparing across diseases, we wanted to use the same sort of architecture so that the types of metrics that we were outputting could be consistent. We could use the same sort of analysis scripts across all of them, and it would all just be really easy. And StarSim provided the perfect way for us to do that. So I'm not going to go into too much detail about all of the models that we developed, because there are 5 of them and I don't have that much time, but I've kind of just got a quick little snapshot. So for measles, as I'm sure you're all aware, it's a highly infectious disease. It's primarily focused in young children, but can of course affect people of any age. And a main method of management for it is trying to maintain as high a level of routine vaccine coverage as possible so that herd immunity will prevent any outbreak at all. But unfortunately, that doesn't hold true in reality. For cholera, it is a disease which has environmental transmission, where infected people will shed the bacteria. It will build up in water sources, and then when susceptible people interact with that water source, they can become infected. So a key method for management of that is the provision of WASH programs, so safe water sources, improving hygiene and sanitation. But again, that doesn't always work. Outbreaks can happen, and we require a vaccine response. Yellow fever is a vector-borne disease. It's primarily carried by mosquitoes, at least in the case of large urban outbreaks, which is what we were concerned with in this analysis. We ignore the sylvatic transmission route for this. But it also is responded to, well, not responded to, but a management method is via routine vaccination to try and maintain as high a level of immunity as possible. And also an important factor to consider is that, yes, it's vector-borne via mosquito populations, and those mosquito populations can be highly affected by environmental conditions, things like the average temperature or the rainfall in a given year in an outbreak setting. And so we have to consider how those factors would impact our model. Fourth up, we have meningitis, and like I said, Mark gave a fantastic overview for that. And I do want to point out that what we considered, we excluded Men-A because of that fantastic impact from the Men-Afrovac campaign, where very little, there are very few Men-A cases nowadays in the meningitis belt because like that, there's fantastic high levels of preventive coverage for that. So we were focused on the other serogroups, so C, W, X, Y, for this analysis. And yeah, meningitis is a highly seasonal disease focused in that meningitis belt in Africa that Mark's map showed. It can be highly asymptomatic. A large proportion of the population can be infected and spreading the disease without knowing about it, but it can have symptomatic presentation with an invasive disease that can be debilitating and has quite a high mortality rate. And then finally, we have Ebola, which, as I'm sure many of you again know, is a very deadly viral hemorrhagic fever. Transmission occurs via contact with blood and bodily fluids from infected peoples, as well as there is a pathway where if you interact with the body of an infected person who's died recently, you can become infected. And so the management and safe burial of corpses is a significant part of the response, as well as contact tracing, quarantine, and isolation, all very key parts of Ebola response, particularly because the Ebola vaccine was only developed quite recently. It only got licensed in 2019. So they were heavily reliant on contact tracing, et cetera, before the vaccine came along. So that's just a quick overview. And I mean, I've skipped over a lot, but if anybody's interested in specific questions about the models or the methods, then feel free to ask me. So I'm going to hop straight into the results because I kind of have to get through 5 diseases worth. There'll be some common themes and you'll see this type of plot a lot. So I'm going to explain it a bit now. So this is the first set of results, which is for cholera. So this plot is showing our set of outbreaks that we calibrated to, and it's showing a proportional reduction in cases and deaths in those 2 columns, cases on the left, deaths on the right. So we've got 2 sets of error bars, and they're basically saying that when we compare our baseline scenarios to our counterfactual scenarios with no ORI, and then we use the counterfactual outcomes as the denominator, this is the proportional reduction in cases and deaths for each outbreak estimated to be due to the presence of the ORI response for these outbreaks. They are color-coded and ordered by response time. So that ranges from around 30 days to around 130 days. They're just sort of grouped in those categories. And what you can see by response time, I mean the time between outbreak declaration and when the vaccines actually start to be delivered to the population. And you can see there's a very clear trend as we move down the plot where with faster response times, the average impact is greater, and with slower response times, the average impact is less. And of course, that makes perfect sense, but it's nice and clear to be able to see that outcome come out from the model. So as I move forward, again, there'll be a few plots that look like this. So I'm going to, now that hopefully you have an understanding of how to read them, I'll skim through it a little bit faster, but I'll make sure to call out key details as we go through for each disease. So this is the similar plot, but for measles. One key feature to point out in this plot is there is an additional grouping. You can see it in the vertical dimension where we've grouped the outbreaks by baseline vaccine coverage. So as I pointed out, having routine vaccination as a management strategy is key for measles. And so we've, again, colored and ordered the outbreaks by response time, but they're grouped within the estimated baseline coverage occurring in the setting of each of these outbreaks. So what we found was typically we're seeing an estimated higher level of impact from the ORI in outbreak settings with less baseline vaccine coverage, which makes sense because a greater proportion of the population is susceptible. So if you can get in and protect them before the outbreak is really taken off, you will see more benefit. But if, of course, there's a really high level of response, like close to 100%, as you can see down the bottom, we see very little impact because there aren't that many people who need additional protection from an ORI response, and most of those outbreaks tend to be relatively small. as we can see in sort of the middle group around that 80%, there's a really nice clear trend of faster responses producing typically greater impact again, ranging this time from 30 days to about 220 days. And then one call out that I have as a really good example of this, I think, is there was an outbreak in the Democratic Republic of Congo in 2021, which occurred in a low baseline vaccination setting. So you kind of naively expected, based on the other results, that we would have seen quite a high level of impact, but we see almost no impact. And that's because in reality, this response took a really long time, about 220 days. And so in the model, we were seeing that, our baseline simulations with a response were producing outbreaks of about the same size as the counterfactual without a response, because it was just taking so long. Most people would be infected before the response could even start. So this is just really reinforcing the importance of, one, maintaining high levels of coverage so that outbreaks can't get that big, and when there are gaps, to respond as quickly as possible. Here we have our plot for yellow fever, which looks very similar to measles because we've again grouped it by baseline vaccine coverage. But an important thing to note for this one is that the color coding is not for response time. In this case, we've color coded it according to transmission level. So what I mean by that is, as I pointed out, yellow fever is a vector-borne disease, and the mosquito population can be affected by environmental factors like temperature and rainfall. And so we included this in the model as either an increase or a decrease to the transmission rates from the mosquitoes to the humans based on the estimated suitability of these factors in a given outbreak setting. So if there was better temperatures, more rainfall, then we would assume higher transmission, and the opposite to hold true as well. And so what we found for yellow fever was that when these transmission rates are higher, we see more impact from the ORI. And again, that makes perfect sense. Higher transmission rates means that a greater proportion of the population can be affected. And so if you're getting in and vaccinating them, then that's just more protection ahead of them getting infected. Interestingly, we found that in low transmission settings or settings with relatively high baseline vaccine coverage, we didn't actually observe much impact from response time because those outbreaks didn't tend to get that big and it wasn't spreading that quickly. But in cases where the transmission rates were sort of medium to high and there was relatively low baseline vaccine coverage, then we again found this relationship between response time and impact. So this is the consistent theme that we found through all of this, is that responding as quickly as possible when there is a high level of risk is always much more beneficial. Now, my final set, I've grouped meningitis and Ebola together because we had relatively fewer outbreaks to calibrate to, so they just fit more nicely on the slide rather than having two very bare sets of slides. But I'll go through them individually. So meningitis in the figure up the top. In this case, they're again grouped, but this is by the type of vaccine that was used. So Mark mentioned that from 2025, there's going to be this pentavalent conjugate vaccine, which will be introduced. That's hopefully will lead to MenA elimination. In this case, it was a quadrivalent conjugate vaccine that was used in some outbreaks. And then in other outbreaks, there was a polysaccharide vaccine. So that has been around for a bit longer. And it doesn't provide the same level of protection against asymptomatic disease as the conjugate vaccines do. And so I've just grouped them by the type of vaccine that was used in each outbreak. Again, these are color-coded now by response time. And we again see this trend of decreasing impact as response time increases. It's a little bit less pronounced here than, say, we saw in cholera and yellow fever and measles. And I think that's due to the fact that meningitis is highly seasonal. It will occur in the dry season in the meningitis belt. But outside of that, the rates of transmission and the presentation of invasive disease are quite low. So even without a response, the model would kind of have a hard cutoff and the outbreaks wouldn't grow to be super big most of the time. And then down the bottom, we've got our small set of outbreaks for Ebola. Again, this is quite limited because the vaccine is quite new. So we have 7 outbreaks that we worked with that we calibrated to since 2020. Sorry, since 2018 is the earliest one. And unfortunately, from such a small set, it's difficult to draw any conclusions about the impact of the response time, particularly because in this set of outbreaks, the response time is very tight. The fastest response occurred within 4 days, and the slowest response was still happening within 14 days. So that just really, talks to how seriously Ebola outbreaks are taken. But it makes it difficult for me to try and draw any conclusions like I had before. I also want to point out that, as I noted, contact tracing and quarantine and isolation are obviously key components of Ebola response before the vaccine came around, and have still continue to be. So we included those in our model. Even without the ORIs or in our counterfactuals, the contact tracing still has quite a strong impact on reducing the size of the outbreak, because that is what has happened historically, and it wouldn't be fair to assume that that isn't going to happen just to make our counterfactuals look bigger. Now, the second type of result that we produced is what I mentioned before. We wanted to consider what impact the ORI would have on the expected size of outbreaks that are observed. I'm only going to discuss measles here in depth because I don't have time to go into everything, but we produced these types of plots for all 5 diseases. And basically what it's showing is I've got 2 sets of bars there, and the top one is showing the baseline distribution for 51 measles outbreaks as they exceed certain case thresholds. So that's ranging from those which are smaller than 500 cases to outbreaks which were larger than 100,000. So just this really huge range of outbreak sizes that we had in our data set. And what we found was that in this baseline, so what happened in reality and what we calibrated to, about 10 of those 51 outbreaks were quite small, less than 500 cases. So that's about 20% of them. And 4 of them were very, very large, over 100,000 cases. So that's less than 10%. But when we ran our counterfactual simulations, and so for those 51 sets of measles outbreaks, when we didn't include an ORI, we found that 8 of them were still below about 500 cases. They remained quite small because they occurred in high vaccinated settings, so there wasn't much room for the outbreak to grow. But the number of them which grew to exceed 100,000 cases was now 21. So it went from fewer than 10% of the outbreaks exceeding this really high threshold to more than 40% of them. So we're finding that the presence of the ORI is really having this strong impact on stopping the outbreaks from growing to be very, very large. And that was that's a consistent finding that we had, that the presence of ORI across all of these diseases is reducing the expected size of the outbreaks, and for some of them, particularly measles, cholera, and yellow fever, it was reducing the expected number of them, which would grow to be just very, very large. So a really nice sort of takeaway from this, I think. And now my final results slide, that I'm not going to dwell on this because it's just a lot of numbers, and I'm not going to go into each one of the diseases because I don't have time. But because a lot of what I've been talking about is proportional impacts, changes in the distributions and the number of outbreaks which exceed certain thresholds, I think it's nice to have some sort of absolute numbers to think about the scale of what we've estimated here. So cumulatively, across 210 outbreaks of these 5 diseases from 2000 to 2023, we're estimating that the presence of ORI has averted 5.8 million cases, 327,000 deaths, $14.6 million, and $31.7 billion in economic costs. So a pretty significant impact, I think. And it was a really nice sort of finding for us to present to Gavi. So as a summary, these really fantastic impacts that across 5 really prevalent diseases in low and middle income countries, the presence of ORI has averted 5.8 million cases, averted $31.7 billion in economic costs. And our really consistent findings across all of these diseases was that the faster you're responding, the more impact you're going to have with these programs. For diseases where routine vaccination coverage is the norm, maintaining high levels of routine vaccination is really impactful as well. It means that there's less work for the ORI to do if an outbreak occurs. And then the presence of ORI is consistently reducing the expected size of outbreaks, and it's reducing the risk of observing these really large outbreaks. So yeah, that is everything. I've probably talked way quicker. I feel like I'm talking very fast, but I'll just take a second to say thank you to everyone who I worked with on this. Of course, the analysis would not have been possible without my co-authors, and then I've also got a huge selection of people to thank. I don't know if Elizabeth is in the room, but there are a huge group of people from a range of different organizations who helped us a great deal by providing expert feedback and some data to us as we developed these models, because it was the first time we'd worked with a few of these diseases. And yeah, I'm happy to answer any questions. And if you're interested, we've got a preprint available, so that is accessible by the QR code. Thank you.
I'll start with a quick question. What information did you have on the coverage rates for each of those?
Yeah.
Break response. Sorry if you mentioned that already. And then also like the speed at which they were scaled up.
Mm-hmm.
And did that matter?
Yes. So great question. So by coverage rates, do you mean the coverages achieved by the ORI? Yeah. So something pointed out by Mark was that for meningitis, Ghana has like 98% coverage from the MenAfriVac. So we're seeing high levels of of coverage for other programs related to meningitis. Some of the measles data that we got from Gavi indicated that they were achieving 90 plus to 100% coverage in the response. And then for some of the others, so I don't think we had any data for yellow fever or cholera, but we because of that, we assumed relatively high achievement rates, like 90 plus levels of coverage achieved. And then for Ebola, we assumed 100%, because we assume it's taken very seriously and they try to get everyone in the area where the outbreak is occurring.
Okay, thanks.
Did you have any data on what the ORI programs cost? Because I saw that you saved $31 billion, and I'm assuming that's more than what the ORI programs cost. Mm-hmm. By how much?
Yeah, no, absolutely fantastic question. And I wish we did. Sort of when this project was being developed with Gavi, we kind of hoped that we'd receive some costing data so that we could do a cost effectiveness analysis of some sort. But unfortunately, we didn't really. We got a little bit for measles, but it wasn't enough for us to be able to look at that, unfortunately. Yeah, it would be great if someone with that data could take some of these results and do it. But yeah, unfortunately, we couldn't.
Thank you for the excellent talk. I'm interested in how you estimated your daily. The DALY. Mm-hmm. So from my understanding, you manage the YLL and DLY, and perhaps also the life expectancy for different age groups. Mm-hmm. So can you perhaps just explain a little how you estimate that, and then also your data source?
Yep. So for each disease, we took the disability weights from the global disease burden estimates, and we took estimates of sort of the different rates of presentation of different outcomes of the disease. So whether it was more severe disease, whether for meningitis, it was causing different severe outcomes like blindness or any of the other presentations. So we weighted for each disease, for those who survived by those disability weights. And then in order to estimate the years of life lost, we took the life expectancies by age from the UNWPP estimates and mapped those to the year and country in which each of these outbreaks occurred. And so the agent-based model as well that we used, you can input age distributions for the populations where outbreaks occur. So we would have estimates of, this is the average age of death in this model in this outbreak. And so we would compare that to the life expectancy in the country where that occurred at, 2015 or whatever the year was. I hope that answers, everything.
Can you tell us more about calibration and data at the scale? So is each dot and whisker its own calibration target? Is there pooling? How did you assemble the data set? It's quite a large scale, and it'd be interesting to understand how you approach it.
Yeah, no, fantastic question. And I really wanted to be able to talk to that, but it would take way more than the 15 minutes that I have. And yeah, you're completely right. So we had a huge selection of outbreaks across 5 different diseases. And so calibrating to each of them individually would not have been tractable. So we basically did what we refer to as a collective calibration approach, where for each disease we had our set of N outbreaks, and we sort of mapped them by a couple of different parameters. So for each of them, it would be by the response time and the vaccination rate. And I realize, Roy, I didn't answer your question about vaccination rates. I'll respond to that after this. So we kind of created like this dimensional lattice, and we would map each outbreak to the best fitting point on the lattice by those 2 dimensions. Some would have other dimensions. And then we would run our model simulations across this lattice. So we would end up like this 2 or 3 or even 4-dimensional space of model simulations that fit not exactly to what occurred in a given outbreak, but close enough. And then we went through like a filtering process, basically where we did trajectory selection, and we only selected the model simulations that were close to a given outbreak, and rejected the others that didn't actually look like what happened in that outbreak. So it was still very computationally heavy, but it allowed us to sort of simplify the process, if that all makes sense. Cool. And then Roy, so vaccination rate. We had some data from like WHO reports and et cetera that would give us the duration of the response, and we'd have our cumulative number of vaccines that were delivered over a given response. So we just assumed a linear rate of delivery of N vaccines over M days to give us a daily vaccination rate. And I think you asked how impactful that was. We interestingly, because most of the that duration data that we had was pretty quick. Like, it blew me away how quickly millions of vaccines could be delivered in some of these outbreaks. So pretty consistently, we saw that vaccines were being delivered very rapidly. And so even though there was variation across the diseases and across each outbreak within a disease, it didn't actually have that much impact because it was always just very, very fast. Yeah.
Okay. Thanks, Tom.
Thanks.